I'm Dr. Andrew Norman, and I've been on the faculty at the University of Cincinnati College of Medicine since 1986. I'm a pharmacologist trained to study how drugs and medications work, particularly in the brain, and I've been invited to talk to you about my development of a unique human antibody against cocaine as a potential treatment for cocaine addiction. Cocaine addiction represents a major unmet medical need, and the NIH and the National Institute on Drug Abuse, NIDA, has designated this disorder a top priority. Other addictions, such as for opioids, alcohol, tobacco, and nicotine, all have approved medications for the treatment. For example, the Food and Drug Administration, the FDA, has approved methadone and buprenorphine substitution therapy and the blocker naloxone for opioid abuse, a nicotine gum and patch for smoking cessation, naltrexone and antabuse for alcohol abuse, amongst other medications. In contrast, for cocaine abuse, no medications have been approved by the FDA. This is despite many clinical trials in cocaine abusers over the last 5 decades that have tried a range of different classes of medications. None have been shown to be effective. In response to this unmet medical need, NIDA established a network of centers around the US to specialize in conducting clinical trials to test novel medications for cocaine use disorder. I was part of a team that established a center at the University of Cincinnati College of Medicine in the mid-90s that successfully competed for substantial federal funding to be one of the original 5 national centers, and this continues to get today. The pharmacology of cocaine is well known, and there is a consensus that dopamine signaling in specific parts of the brain mediates cocaine abuse. There are many medications that target dopamine signaling, but as I said, they don't seem to work in the clinic. So while setting up these national clinical trial centers, several panels of the top experts were convened by the government to advise on which medications should be prioritized for clinical testing. Surprisingly, these panels could not come to any consensus. Obviously, there is a fundamental knowledge gap in the understanding of cocaine abuse that continues to impede progress in medications development. It is widely believed that cocaine is addictive by being positively reinforcing, by producing intense pleasure. So animals and people like it, and so they keep wanting that pleasure. However, it is difficult to measure liking and wanting scientifically. I believe that it is this unscientific approach to understanding addiction that results in several paradoxes that are responsible for impeding progress in medications development. Indeed, addicts often report that they don't really like taking the drug, so that is difficult to reconcile with this pleasure-based approach. In contrast to the currently dominant psychological approach to cocaine addiction, I am inviting you to look at cocaine addiction from a new perspective. Cocaine is a drug, and it should exert its effects according to the principles of pharmacology. Drugs that activate a system induce a response, and the magnitude of the response depends on the level of the drug at its site of action in the body. Cocaine indirectly activates dopamine systems in the brain. So, if the cocaine concentration could be reduced there, its effects would also be reduced. One of the strategies that came up during the panel deliberations was reducing the cocaine concentrations in the brain by immunizing addicts with a cocaine vaccine. Another approach that I decided to take would be to generate so-called monoclonal antibodies. You've heard recently about cocktails of monoclonal antibodies that with one infusion can neutralize the SARS-CoV-2 virus and treat COVID-19 pneumonia. We can use the same approach to neutralize cocaine. And my team has used a lot of state-of-the-art science, including vaccine development, transgenic mice, protein chemistry, and genetic engineering to generate and then develop a unique humanized monoclonal antibody that specifically targets cocaine. animal models of human diseases are essential for developing new medications and treatment strategies. We found that pre-treating mice and rats with our antibody stopped cocaine from getting into the brain by keeping cocaine in the bloodstream. In humans, cocaine use disorder is characterized by periods of sobriety and then repeated relapses. So relapse prevention is the overall clinical goal. It would be nice to have animal models of this. Rats also readily acquire cocaine addiction-like behavior, which is a good model of cocaine addiction in people. Rats are placed in a chamber, and every time they press a lever, an intravenous dose of cocaine is delivered. Although they have unrestricted access to cocaine, the intervals between injections are very regular at a particular dose. When cocaine access is stopped, they press the lever for a while and then eventually stop. Then they behave like normal rats. The next day, when these normally behaving rats are placed back in the chamber, they sometimes will go and press the lever. This may be a model of environmental cue-induced relapse. But if no cocaine is delivered, they press a few times, then quickly stop. So this is a very transient phenomenon. However, it has been known for decades that if a dose of cocaine is given to these rats, they quickly begin lever pressing and will persist for some time. This is called priming and is recognized as a model of relapse. My laboratory devised a computer program that controls a pump to gradually increase the cocaine level in the rat's body until they start taking cocaine again. And we call this cocaine level the priming threshold. I predict that anything that increases the cocaine priming threshold in these rats will decrease the probability of maintaining a relapse event in people with cocaine use disorder. When these rats are treated with a single dose of our anti-cocaine antibody, the level of cocaine that will reinstate cocaine self-administration is substantially increased. And this effect persists for several days before it gradually wears off. This, and the data showing that cocaine is kept out of the brain, predicts that a single dose of the antibody will be effective in decreasing the probability of relapse. And because the antibody is eliminated from humans more slowly than in rats, the effect of a single dose in humans should last for about 3 weeks. Using genetic engineering, we have manufactured large amounts of antibody protein pure enough to go into humans. With this material, we completed all the tests required by the FDA to show that it is likely to be safe in humans at the dose range that we believe will be effective. These tests include binding to human tissues and short and long-term toxicology studies in male and female rats at much higher doses than will be used in people. There was no evidence of binding to human tissues or toxicity. We've filed an investigational new drug application to the FDA to allow us to proceed to first-in-human clinical trials. The FDA has requested additional information before we can proceed, and we are in the process of providing that information. I've devoted more than 20 years of my career to this project, and I am determined to complete the first-in-human clinical trials to see if it is safe for clinical use before advancing the antibody through clinical development and pursuing commercial licensing. I invite you to follow our continuing journey to a day when we will have an effective pharmacological solution to the devastating problem of cocaine addiction. Thank you.